I'm going to talk about some work we're doing to develop better model systems to understand pancreatic cancer. The problem with the type of research we do, or one of the problems with the type of research we do in the lab, is that the cells we use and their behavior in the sort of dishes that we grow cells in, it just doesn't really look that much like pancreatic cancer. Now, don't get me wrong, these cells are from pancreatic cancer, they are pancreatic cancer cells, but it may be no surprise if you grow a human pancreatic cancer cell on a piece of plastic in a dish in a lab, it doesn't behave massively like pancreatic cancer. But I mean, what experiment can you do? I've got to do an experiment. I've got to use something as a system to understand. So it's the best we've got, but it's not perfect. And I mean, there are other systems available. So I think I've listed there that you can, if you want to, grow human cancer cells in mice, and you have to destroy their immune system, else they would just recognize the human cancer cell and destroy it. And that's a different system to work on. Is that better? Well, it's probably not better, it's different. But I mean, no surprise, it's a mouse, it's not a human, it's not going to be the same, and it's a human cell in a mouse, that's even weirder. It's not going to be the same. And maybe the final iteration of that is that you can grow mouse cancer cells in mice, but they're mice, and actually mice don't get pancreatic cancer, really, so you have to do some strange stuff to make that happen. So these are the model systems we have. We've got to do experiments using something, but they are not perfect. So that's a problem. What's the solution, or what is a solution? So, I mean, the top statement, it's kind of obvious, isn't it? We need to be growing cancer cells from patients in a more realistic environment rather than in a plastic dish, in something that vaguely resembles the environment that they'd normally be in. And then they're going to behave like pancreatic cancer cells should do, and therefore we can understand their biology and understand how to kill them, which is really what we want to do. So I'm going to tell you about 3 ways that we've done that, not in huge detail, so this isn't going to go on for very long, I hope. Well, there they are. Patient-derived organoids is one technique to try and encourage pancreatic cancer cells from a patient to grow in a more realistic environment. I'm going to mention fibroblasts because I can't give a talk without mentioning fibroblasts. That's what Lewis is working on. And I'm also going to talk about some explant microfluidics cultures, which are maybe next level sort of model system if we can get the things to live for long enough.